All right, ladies and gentlemen, welcome back. We're going to continue our discussion on utilizing characteristics as a way to identify or at least hypothesize the evolutionary relationships between organisms. And today we're going to talk a little bit about their genetic structure, their genomes, as this has become really the sort of first go-to in terms of data for determining those evolutionary relationships now that genetic sequencing has become much more reliable, much cheaper, and much faster. That being said, we still use the morphological data, the fossil record data, as a way to continue to support this genetic information, but this tends to be the first thing we go to now. So what we're talking about here really is comparing either their nucleic acid data, so that could be DNA or RNA, or other molecules, usually proteins, so amino acid sequences, to infer the relatedness um, is a really valuable approach because it's much more subjective as opposed to objective when we're looking at physical characteristics. Um, those physical characteristics can be interpreted in different ways depending on how close one trait is to another when you're talking to different people. So this is pretty much numerical data. We can count the number of changes, and so it's a much more objective approach. So when we look at DNA, right, that DNA codes for different pieces of information, things that code for things such as rRNA are relatively slow. And so that particular set of data can be really useful when I'm looking for branching points hundreds of millions of years ago. Mitochondrial DNA, on the other hand, evolves relatively rapidly. It also only gets passed from mother to offspring, and so it can be much more useful for recent evolutionary events. So just like anything, the type of molecular data that we choose to look at can have more value in one situation as opposed to another. And those are just two examples of that. Regardless of what we look at, we'll often look at gene duplications. Um, Gene duplication events usually occur in a mistake in crossing over when we go to make gametes. It's not the only way that they happen, but it's a common one. Regardless of how they occur, it ends up increasing the number of genes in the genome of that resulting organism, and if it ends up becoming beneficial enough, then ultimately increasing the genome size of the offspring, the species as a whole that diverges from that point. So those gene duplications really provide for more opportunities for evolutionary change. Repeated gene duplications end up resulting in this thing we call gene families. So they'll duplicate the same general location, but then they may take on different traits, and those duplicated genes can be traced back to a common ancestor. So when we look at these gene duplications, they end up fitting usually into two types of homologous genes. Either this idea of orthologous genes, where I find a single copy in the genome, but there's been a change within the gene itself that is homologous between species. So here's my ancestral gene. I had some sort of gene mutation that occurred within that particular sequence. And now these genes are considered orthologous to each other in these two separate species that now have diverged after a speciation event occurred. Paralogous genes, all right, are what we're looking at over here, where it actually forms within the species itself, but it is duplicated and mutated at once, all right? So this is just sort of a linear change in that species over time. This would be a true branching point we would see in the cladogram or phylogenetic tree that we would be building. So orthologous genes are widespread. They are extensive across a lot of different varied species. We see examples of orthologous genes, just for an example, humans and mice. We use this orthologous mutation to see that we diverged somewhere around 65 million years ago, and about 90% of our genes are orthologous, meaning we have a hemoglobin gene, we have a gene for lactase, for example, but the actual gene sequence of that hemoglobin, for example, has certain differences within them. So although we carry the genes for the same traits, the DNA sequence for that trait has diverged. Gene number and complexity of an organism isn't necessarily tightly linked, so just be careful with that. A lot of students in particular assume that the higher level organisms must have a more complex genome, and the way that we express that information might be true, but the actual number of genes that we have, not necessarily. So, for example, humans only have 4 times as many genes as a yeast, and yet we would assume because we are much more complex of an organism than yeast is, which is just a little ball of fungus, that we must have huge numbers of genes comparatively speaking, and that's not necessarily true. We are just much more versatile in the way that we use our genetic information compared to something like a yeast. So each gene can actually perform multiple functions. We'll talk about that when we get to gene expression this year, where the yeast, it's pretty much that gene does that job and that's it. So scientists have looked at this idea of molecular clocks. So can we look at changes in the genome as a way to trace back actual time? So yes and no. We have to make some assumptions here about how often genes change over time. We think that there's some consistency in the rate that those genes mutate, um, and so we can use this to a certain degree. Something like orthologous genes, those nucleotide substitutions are typically proportional to the time since they've occurred in a shared common ancestor, where the paralogous genes, the nucleotide substitutions are proportional to the time when the gene duplicated. But we can use these to calibrate those branches when we know other data. So this is just one piece of the puzzle. Um, there are definitely problems with it. They don't always run smoothly as this neutral theory predicts. And neutral theory just states that much of the evolutionary change in genes and proteins would have no effect on its fitness and not be influenced by natural selection. So that would assume then, if we say that that's true, that those molecular changes in their genes and proteins would be regular. All right, kind of like what we would expect a clock. But if you know anything about clocks, even some clocks seem to run a little fast or a little slow, and that's kind of true here too. There are definitely irregularities that result from natural selection in those DNA forms or those DNA changes. Their estimates of evolutionary divergence that are older than the fossil record certainly have an even higher degree of uncertainty, and the use of multiple genes may improve this estimate. So if I can see that a DNA change happened in this gene, and I expect it to be this old, and then I go to 3 or 4 other genes and I get a very similar result, then we can have a little more confidence in saying that this is in fact working the way we think it does. So it's like anything else. It's an idea, it works, but there's always some minor exceptions to it, and to the more data that we can use to support our hypotheses here when building these trees using this molecular data and this idea of these molecular clocks, the more confident we can be in that idea.